Today we have Dr. Floyd Nichols, who is currently a postdoc at Virginia Tech, although he just informed me he's going to be starting a tenure track position there in August. So it's, yeah. So he will be recruiting. Yeah, so he's a geochemist and astrobiologist. His research focuses on studying biological signatures and also reconstructing paleoclimate and paleohydrology or paleohydrologic information from hypersaline environments in particular. And he does that by using organic geochemistry, computational methods, particular machine learning, statistical techniques, and also remote sensing. I saw you have a couple of, at least one undergrad using remote sensing. He has a bachelor's in biology from St. Vincent College, and then a PhD from the Earth and Planetary Sciences Department at Northwestern University, where he finished in 2024. And he started at Virginia Tech in 2024. And like I said, he's going to be starting a position there soon. And I actually came to know Dr. Nichols because we're in a proposal together, even though we've never met each other before. But he does work in an environment up in British Columbia that one of my former PhD students and current postdoc, Dr. Emmy Hughes, has been working on. And I know she's interacted a lot with him on that. And so I'm really excited to hear about your work in hypersaline links. So yeah, thanks for coming down.
Thanks so much. Thanks for inviting me, both you and Emmy. I'm really excited to share a little bit about my work. As Francis has already mentioned, my work kind of spans a lot of different things, from thinking of things from an astrobiology perspective to paleoclimate, and also implementing computational things like machine learning. And so for today, I'm really going to focus on some of the current work that I've been doing, which is using machine learning approaches to understand and examine biosignatures in planetary analog environments. And so the research motivation for today's talk is really three main points, which is to improve our understanding of potential habitable environments on Mars and perhaps elsewhere as well, as I'll get into a little bit later. And also with these analog environments, specifically with hypersaline systems, there's often these complex and nonlinear interactions between organics and perhaps other environmental parameters and geochemistry. And so trying to unravel those interactions and understand the influence of particular parameters on these organic signatures is something that I'm really interested in and part of the main research motivation. And then, as I sort of alluded to just briefly, is sort of bridging the gap between data science and astrobiology, because especially in the modern time, sometimes there's this disconnect between machine learning and AI and perhaps how we can use it for scientific work. Sometimes it can be used for really good insights, but other times it might not actually be all that useful. So hopefully I can, throughout this talk, share with you how certain aspects of machine learning can be used for different types of problems. And so the main overview for this talk is, first, I'll give you a little bit of background and motivation for some of the research work. So I'll try to talk a little bit about the organics and what they might mean, and a little bit of the machine learning as well, but I'll try to keep it very high level for everyone. And so if anyone does have any questions, though, please feel free to stop me if something doesn't make sense. And I'll also talk about the varying applications of machine learning for astrobiology, specifically in these analog environments. And then just thinking about some broader implications. And so when I say thinking about the varying applications of machine learning for astrobiology, as I was thinking of about putting together this talk, I was wondering, how do I want to sort of express what I mean by this? And so what I decided was to sort of divide this talk into like three main parts, really, which is going from thinking of how machine learning can be used or has been used, specifically with my work, on smaller data, which oftentimes we think that machine learning can't necessarily be used on small data, but depending on the type of method that you're using, it actually can, and it can be very effective as well. And then sort of working our way through potentially bigger data. And tangentially, with some of these small data sets are often, quote, simple. And the reason why I say quote is because As many of you who do research know, most data is not simple to begin with, but we'll just use simple for this classification. And in this regard, we'll think of it as sort of unimodal data. So data that is just one mode of collection. And we'll increase the complexity throughout the talk to then discuss more complex, maybe multimodal and multi-scale data and how machine learning might potentially be used for assessing biosignatures in these more complex data. And so to start with some background, I'm sure many of you already know in this seminar series, but in case those who don't, I'll give you a brief background on sort of astrobiology, why we're interested in, and some potential astrobiological targets. So astrobiological targets, you can really think of as planetary bodies that are of great interest to scientists for having potential, has hosted potential life, either in the past or in the modern. And so some of these astrobiological targets in our solar system are icy planets or icy moons, such as Europa, Enceladus, Titan, and our favorite Mars, or I should say my favorite Mars. But I guess it depends on who you ask. If anyone studies Europa here, please don't beat me up. I do also think about Europa as well, which I'll briefly describe later in the talk. But these are some of the main astrobiologic targets in our solar system for many reasons. But the main reasons are that these particular planetary bodies either host large amounts of liquid water, such as beneath these icy shells of Europa and Enceladus, and perhaps even more unique types of hydrologic systems like Titan, which is mostly methane rich, very cold, so methane is actually liquid. And then Mars, which is also perhaps an ocean world. Although perhaps you're looking at this image of Mars and you're thinking, I do not see how that is an ocean world. That looks like a completely desiccated landscape. And you would be correct. It kind of is in the modern. However, Mars once had vast oceans on the surface of the planet, but through time, as it lost its atmosphere, it also lost the water on the surface. And so now what we see in the modern is this more desiccated landscape. However, with Mars having hosted liquid oceans in its past, that does make it very attractive for thinking about potential life to have arisen on the planet of Mars. And more precisely, Mars lost its atmosphere right around the same time that we see onset of life on Earth. And so the question begs, can life have arisen at the same time on Mars? And so one of the main goals for a lot of astrobiological studies when we're looking at these planetary systems or analog bodies is sort of going back into the past and thinking about the signatures that we see. However, unfortunately, I don't believe anyone here owns a time machine, so we can't necessarily go back in time to investigate these environments. However, we can understand some more fundamental principles that might be associated with organics, organic preservation and production, and similar types of environments. And we call these environments analogs. In this particular instance, we'll call them Mars analog environments, but in cases where we might be thinking of the icy moons, and we might think of them as just planetary analog environments. And just to give you a few examples of different type of analog environments on Earth that host a variety of chemistries and environmental parameters. You can see in the top left, there's Yellowstone hydrothermal springs, which many of you may be familiar with, but it's a very hot system. And we also have the Atacama Desert in Chile, which is one of my favorites, most recently at least, which is a very hyper-arid desert that also has a ton of radiation. So it's actually a really good analog for thinking about the surface of Mars, modern Mars. Then you also have lava tubes. Here I have depicted the Hawaii lava tubes, but these are areas that don't receive a whole ton of sunlight. And so the energy movement through these systems is going to look very differently than how we might think of on the surface where you can have photosynthesis driving a lot of that energy. Then you also have Don Juan Pond in Antarctica, and also there are other hypersaline lakes in Antarctica, but not only are they hypersaline, but they're very cold too. I don't think I'd want to take a bath in that. And then finally, again, these are just a non-extensive list of examples, but there's also the Dala hot springs in Ethiopia, which is this very, very sulfur-rich, acidic, and also very hot system. And that's actually why you see this yellowish color is mostly from sulfur precipitation. So when you look at these images, you might think that they were on a different planet, but they're in fact here on Earth. And that's actually what makes them so attractive for studying the potential for things such as biosignature production and preservation, or perhaps some other astrobiologic relevance as well, such as what you might see with remote sensing. And so for this talk, I'm going to focus on two main environments, which is where a lot of my research work in grad school and currently is being performed. And that's going to situate really the rest of the talk. But as I mentioned, the Atacama Desert in Chile is one of my most recent favorite Mars analog environments. But the Atacama Desert is in northern Chile, as denoted by this white rectangle. And as I mentioned previously, it's a very arid desert. It's been arid for a very long time, on the order of 1 million years, and it's actually one of the longest dry deserts on Earth, not really counting Antarctica. And it also has a ton of radiation at its surface. And so this makes such a great modern analog for Mars. And so trying to understand the biosignature distribution and organic molecule distribution in these environments might help inform what we might potentially see on the surface of Mars if we ever do detect any biosignatures. And then another system that I work in that's a little bit different, actually very different, are the Caribou, Hypersaline lakes of the Caribou Plateau, which a couple of people in this room might be familiar with. I won't name names, but the Hypersaline lakes of the Caribou Plateau are very interesting because, well, they're kind of in an unassuming location, right? You would not think British Columbia would host these plethora of hypersaline systems. And in fact, they're not only hypersaline, but they have weirdly unique chemistries. And so for those of you who might not be familiar with hypersaline systems, generally, most of them on Earth are sodium chloride dominated. So you think of table salt. But here in British Columbia, we have lakes such as Bass Lake, Salt Lake, and Last Chance Lake that have their dominant chemical compositions being magnesium sulfate and sodium carbonate. And this unique chemistry is actually perhaps unique on Earth, but not so unique on Mars. We know we've seen many magnesium sulfate deposits in different craters on Mars, and also tons of carbonates. And so despite these chemistries being vastly different than most of the things that we see on Earth, they actually might offer a window and or natural laboratory towards studying some of these interactions that we might see with organic molecules. And here I'm just showing you some images of these particular lakes that I work in, so Bass Lake, Salt Lake, and Last Chance Lake. And I won't really talk about it in this talk, but you can see in the Bass Lake, Salt Lake, and Last Chance Lake, they have these weird appearances. We call them spotted appearances, and we don't really know why they formed, but perhaps someday somebody in this room might help us understand why they form. But one of the reasons why I like to show these images is because if you ever go there, if you have a chance to go there, it really does feel like you're on a different planet, especially with seeing these different spots, which you can't really see for scale in these images, but some of these spots are maybe like a couple meters in diameter, especially at Last Chance Lake, and in some cases, Bass Lake. But the Atacama Desert provides a good modern analog, and these hypersaline lakes from the Caribou Plateau provide a better or perhaps more relevant ancient analog for the surface of Mars. Okay, so I keep throwing out the term organics, organic molecules, biosignatures. What do I really mean with that? Here, hopefully, I'll walk you through again, just very broadly, a broad overview of the different types of organics that we can use to assess potential of production and preservation in a particular environment. And so more precisely, my work generally involves using a combination of organic carbon, lipid biomarkers, and stable isotopes. And so organic carbon, well, organic carbon is a lot of things. So here I'm highlighting this microorganism as it's generally organic carbon, but organic carbon is really going to be any of the material that's going to come from living organisms. So that could be proteins, lipids, nucleic acids, the whole hodgepodge of everything. So we can generally think of organic carbon as our broad pool of organics in a system. But where a lot of my work focuses on, in grad school at least, and even now, is lipid biomarkers. And so lipid biomarkers are these components of the cell wall, which is what I have depicted here, where you have a polar hit group, such as what's defined by this circle, so something like phosphate. So it's very polar, and then you have these hydrocarbon tails here, which are nonpolar. And so looking at this structure, you might think it doesn't tell you too much information, but what makes lipids very attractive are two main things. One is similarly to DNA, lipid biomarkers such as this intact polar lipid that I'm showing as an example here can offer some diagnostic capabilities of microorganisms. So for instance, we can extract DNA from environment and say more or less precisely, maybe this is actinobacteria or something like that. Lipids perhaps aren't as precise as DNA, but what they can say is maybe it's this class of microorganism, or maybe it's this class of eukaryote or something like that. And so although they're not as diagnostic as DNA, one thing that they do perform better at, or I shouldn't really say perform, but these lipid biomarkers can persist in the environment for a very, very long time. So DNA has even more polar functional groups than what we see here from the lipid biomarkers, so they degrade very rapidly in environment. But if we want to study ancient Mars, ancient planetary systems, we probably want to look at a biomarker or organic molecule that can also persist for, let's say, equal amounts of time. And that's what makes lipid biomarkers extremely attractive. And then stable isotopes are really, there's a variety of different types of stable isotopes, but here I'm generally looking at carbon stable isotopes, and that's often the change in the neutron of a particular element. So in this case, you can see carbon 12, carbon 13, and these blue and reds are going to indicate your neutrons and protons. And so if you add a neutron to an element, you don't actually change the charge, but you change the mass. And sometimes this mass is a very small amount, and I won't get into it for this talk, but this change in mass has some implications for kinetic properties. And so thinking of them more broadly, because of these changes, I like to think of isotopes as fingerprints. And so because we can't use lipids to be as diagnostic as DNA, for instance, we can actually pair lipid biomarkers or organic carbon with stable isotopes, such as stable isotopes from carbon, to provide even more information about what we see in the environment and also the environment itself, such as if you're looking at hydrogen, for instance, where you can actually reconstruct hydroclimatic activity. And so these are the three main suites of organics and organic markers that I'm generally looking at. For the first part of this machine learning portion, I'll mostly focus on the organic carbon pool, so broadly organics in a system. And in case there's anyone who's interested, I'll also give you a very, very brief description of some of the methodology. But when we're in these environments, whether it's the Atacama or British Columbia, we can collect a suite of things such as surface sediments, salt deposits. We can filter brines and water and collect the organic matter and other particulates on these different filters. And we can do a process called Bly-Dyer lipid extraction to basically separate out polar things, which is shown in the blue, versus non-polar things, which is shown in the yellow. And again, lipids are generally nonpolar, so it's like a really good way to separate out the components that we don't necessarily want, like proteins or nucleic acids or amino acids or something like that. Then we can do some separation to separate out lipids into more specific classes. So for instance, there are lipids such as fatty acids or lipids such as alkanes or alcohol lipids. Those all have different functional groups. And so sometimes we're interested in looking at one, two, three, or four, and they all tell a slightly different story. And then the main method of analysis that I use for lipid biomarkers is gas chromatography mass spectrometry, or GCMS for short, which basically just allows you to further separate out compounds by basically size, chemistry, and then we can fragment and reconstruct what those molecules actually were. So I often think of it as putting together like a puzzle piece, although when you look at a screen long enough, sometimes it doesn't feel that way. And using these same type of samples, or the same samples really, we can also do organic carbon and isotope analysis. And the nice thing is, because I use what's called an elemental analyzer isotopic ratio mass spectrometer, or EAIRMS for short, I can do both organic carbon and isotope analysis at the same time, which saves a lot of time, because sometimes you're working with like 200 or 300 samples. So it saves mental health too. And so I won't get into the EARMS too much, but just know that this particular instrument can be used to detect, to quantify both organic carbon and the stable isotope composition of carbon, and also other elements too, like nitrogen. Additionally, because I had mentioned time, time is an important aspect that we're often interested in, although in really complicated systems, sometimes time doesn't necessarily mean anything. But we also can do X-ray fluorescence to detect elemental abundances, which can provide some geochemical information about what's happening in a system. So to do that, we'll collect the sediment cores, and we can run these cores through what's called a geotech core scanner. And this geotech core scanner has a variety of different instruments that you can see here, but there's some X-ray, there's also a digital camera, so you can get these nice high resolution images of these cores. And again, we can output information such as the image or also elemental abundances. And specifically with the instrument that I was working with, we can get elemental abundances from magnesium up to uranium. And a little bit later in this talk, I'll try to explain what some of those elements might mean and why we might be interested in the geochemical information provided from those elements with respect to organics. Okay, so now that hopefully you got a background of astrobiology, some of the organics that I work with, and briefly the methodology as well. Now we'll try to shift a bit more into the machine learning aspect of this talk, which is really the sort of the meat and potatoes. And so the first thing I wanted to do is identify a problem. Why would I want to use machine learning? So as you can already tell from these previous slides with these elemental abundances and organics, modern technology has allowed us to get and obtain data really, really rapidly. So we get these large and complex data sets in geosciences, astrobiology, earth sciences. However, if we try to use, let's say, the traditional methods of extracting information or understanding patterns within these data sets, perhaps these traditional methods might fall a little bit short. So we sort of need maybe more advanced methods, or methods that are more effective for analyzing and understanding these large and complex data sets, or larger and complex data sets. And one nice thing is that machine learning is a potentially valuable tool that might be able to tackle some of these problems that we have run into with these increasingly large and complex data sets. And so just to put it into perspective, this is a data sheet that I was working with in grad school, which is big, but not that big when you think about it. But I remember my advisor had asked me to try to understand what was happening with the organic carbon in the system. And I looked at this data sheet, and then I closed my laptop, went into a corner, cried a little bit, and then I came back and I was like, there's got to be a better way. And so that is where I entered my journey of machine learning, all from this spreadsheet. And hopefully in the next couple of slides, I can show you how it actually has proven to be really effective in understanding the sort of relationships and patterns with all of these different data set or all of the different data points here. And so here I'll provide a brief description of machine learning too, for those who might not be as familiar with some of the techniques. And again, this is very broad. But again, the main goal or question is, can we utilize the predictive powers of machine learning? And specifically for these questions, I'm interested again in understanding the biosignature distribution with respect to different chemistries, environmental parameters. And so that's what we want to predict. And so there are two main classes of machine learning. Well, it gets a little bit more complicated than that, but for simplicity, we can say that there's two main classes for machine learning, that being supervised learning, which can be broken down into classification and regression. So classification can be used when you're working with discrete data sets, and regression is used when you're trying to predict something from continuous data. Then you also have unsupervised learning, which can be further broken down into dimensionality reduction and clustering. So dimensionality reduction is when you have a large dimensional data set, such as this one, and you want to convert it into something that's more meaningful with less dimensions. And so that's what dimensionality reduction is. And then there's clustering as well, which is basically identifying groupings within a data set. And so, as I mentioned at the start of this talk, I want to mostly break down this talk into three main parts, that being applications of machine learning with this smaller / unimodal data, and sort of work away with complexity and how maybe we can tackle some of those different questions. So for this first part, I'm going to be referring to the data set from British Columbia, or basically that's Excel spreadsheet that you just saw 2 slides ago. And for this data, the ultimate goal was to predict organic carbon from elemental abundances to see if we can understand any relationships that might be present between organic carbon or life and sort of environmental parameters, elemental abundances. So the first thing I did for this data was I implemented what we call exploratory data analysis, or EDA, using unsupervised learning. And unsupervised learning is really great for understanding the underlying structure of a data. This can mean different things, but for simplicity, one thing that the underlying structure of the data might be is, are there a lot of linear interactions? Are there a lot of nonlinear interactions? And if there's, for instance, say a lot of nonlinear interactions within a data set, then perhaps methods like machine learning can be extremely useful, like supervised machine learning. And just to briefly describe the data that I'm using is we're looking at organic carbon as our response variable, so we're going to predict, and the elemental abundances as shown here as our explanatory variables are basically what we're going to use to inform their predictions. And I'll describe it briefly, but if anyone has questions, I'm more than happy to answer. But for this, the next couple of slides in this talk, I broke this down into a classification problem rather than a regression problem, mostly for simplicity for the model. And so for this classification, basically, we use this histogram distribution to create 3 main classes to incorporate relatively equal amounts of data in each bin. So we look at low organic carbon, which is about 0 to 2%, and then moderate, which was roughly 2 to 10%, and then anything over 10% was considered high in organic carbon abundances. And so the main type of unsupervised learning methodology that I use, and some of you may already be familiar with this already, is principal component analysis. So principal component analysis is a dimensionality reduction technique that can be used to, again, reduce the amount of dimensions in high dimensional data to something that's a little bit more understandable. And so one of the first things I did with the principal component analysis was first looked at the linear PCA. A linear PCA is really what we consider to be the sort of the standard principal component analysis. And you can see just looking at panel A, it does do well with producing some clusters. So the way that you can read these PCA diagrams is things that cluster together have more similarities in their composition. So in this case, their elemental compositions are much more similar when they cluster together than when they're further apart. So we can see some groupings, but they're not super clear in some cases, especially in this region. But then the next thing I did was implemented nonlinear PCA, which is actually a type of methodology that most people don't necessarily implement, but can be extremely valuable when you want to assess and reduce dimensions in a nonlinear fashion. And this can tell you a little bit about, are there a ton of nonlinearity or nonlinear relationships in your data? And so when I did the nonlinear PCA, and again, I can describe it a little bit more, but there's different methodologies. In this particular case, I implemented what's called a radial basis function, which just allows you to reduce your data nonlinearly using radial basis function, but you can also implement things like polynomial functions, etc. And in this particular dimensionality reduction, you can see that the patterns start to show something a little bit different. Not only do we see these clusterings, which are similar to our standard, but we can also start to see sort of a differentiation in the point sizes, which indicate how far along they are in the depth. So we can start to see a little bit more structure from this nonlinear PCA. And I should mention too, one of the nice things with nonlinear PCA, it is computationally expensive, but if your data is very linear and has mostly linear interactions, it will basically plot the same way as a linear PCA. So if they plot differently, and if the nonlinear PCA plots a little bit better, then that tells you a little bit more about the linear interactions. But one thing is when you look at these two in the two-dimensional format, they do look different, but they don't look vastly different. And so the next thing I thought was, okay, how does this look in three-dimensional space? Can we see more structure? And immediately we do. So in panel C, the first thing that I noticed was everything is mostly happening on these two axes, PC1 and PC2. But in the nonlinear PCA, we actually see a lot more structure sort of forming in the principal component analysis. And what these two panels tell us is that, all right, there's a lot of nonlinear interactions in this data set. And that makes sense, especially when you look at the hypersaline data with the organic carbon as they go through with depth. It looks very unfortunately nonlinear, which makes things so complicated when you want to interpret the data. And so basically this PCA was just confirming what we saw visually. So once I had a better idea of what the structure of the data looked like, and more precisely that there was a lot of nonlinear interactions, I was able to use the exploratory data analysis to select a series of machine learning models that I thought would best be used to implement predictions for organic carbon from these elemental abundances. And more specifically, in these supervised machine learning models, I implement four main algorithms, that being k-nearest neighbor, random forest, support vector machine, SVM, and logistic regression. You don't necessarily need to know what these are, though I'm definitely happy to discuss it. But the main implications for choosing these four is k-nearest neighbor and random forest are these sort of non-parametric supervised machine learning methods. So they excel generally when you have a lot of nonlinear relationships and nonlinear interactions in your data, whereas support vector machine and logistic regression are much better at linear data, making distinctions from linear data. And visually, what you can see in these figures here, which are confusion matrices, is the way you read these confusion matrices is you have your predicted labels on the x-axis and your true labels on the y-axis. And basically what you're looking for is for all of your values to fall along this diagonal. So when you predict something at one category and the true label is also that category, that means it's a true positive. So basically, we're looking for really good numbers on the diagonal. And immediately, what you can see is our best performing supervised machine learning models were actually random forest and k-nearest neighbor, which again, generally perform much better with nonlinear data compared to support vector machine and logistic regression. So again, this is actually just kind of confirming what we saw in the principal component analysis or dimensionality reduction. And even though our support vector machine and logistic regression didn't perform as well as random forest and k-nearest neighbor, they actually all generally still had pretty good accuracy, ranging from about 73% to 88%. And I don't have it shown here in this slide, but we also produce what's called a concept model to see how these predictions might stack up against, let's say, just randomly predicting from your data. And in those random or a baseline model, it's about 40%. So these models are actually performing about 40 to 50% better than if you were to randomly make a guess from your data set. But unfortunately, machine learning doesn't always end there, because you can have a good model, but sometimes you might get really lucky for that one instance of a model, which is often has a lot of different hyperparameters. Maybe you got really lucky, or I should say, maybe I got really lucky and got really good hyperparameters from the tuning that I was doing. So to validate the model, I also had to take the model that was used on this elemental abundances to predict organic carbon, to predict to make predictions on a completely new data set. So something that the model has never seen, never been trained on before. And so that's what these figures are here. These panels are essentially showing the probability distribution of a particular class with respect to the sample index, which is not super important. But the main thing from this particular sample data set or sample indices is they were all very low or lean in organic carbon, so less than 2%. And you can see really what we're looking for is this pinkish red zone to have the highest probability. And we can see that the random forest, support vector machine, and k-nearest neighbor, logistic regression, were able to generally perform really well on these new data sets and having high probability that what we saw was low, which is what we actually tested in the lab too, by looking at the organic carbon. But one thing that you can see is the k-nearest neighbor actually kind of falls off a little bit, even though the actual model itself was in the previous slide had really good accuracy. So compared to the other models. So with the validation as well as the model performance in the previous slide, we saw that the random forest was just going to be our best model to use. And so that's what we use going forward to assess these interactions. And one thing that I forgot to mention is these particular models here are what we call more traditional machine learning methods. And what's really nice about the traditional methods is we can actually do feature extraction to understand the interactions that are happening, which is a little bit different than neural networks, although I'll say that in quotes because that's not true for all of them, and I'll show you a little bit how we can assess that. But looking at the random forest, I then implement some feature extraction to assess the importances of the different variables that we see in the data set on the influence or on the predictions of organic carbon. And so I just have this here for reference, but I implement basically three main types of feature extraction. And the reason why I did that was because one, if you're trying to get this through reviewers and they only see one feature importance, they might be like, well, how does this look if you also look at a different weighted system? And so doing all three is a really good way to see if all of the features or variables corroborate each other. And for the most part, they do. And so in this right side panel here, I'm showing the top 5 features that we extract from both or all three of these feature extraction methods, and then did some more exploratory data analysis to see how organic carbon related to each of these particular variables. And one of the most interesting things that I saw from this was we see a lot of elemental abundances that are often associated with things that we might think of as playing an important role for organic preservation and or production, such as iron, titanium, silica, aluminum even. And so these are the elements that are often associated with clays and detrital material. And we know that often is a very important role for organic preservation, mostly because of the absorption of organics to the mineral surfaces of these type of elements or minerals. But one of the most interesting things was I also saw some zinc and uranium, and not in the top 5 here, but also other things like nickel. And I was thinking, why are these so important for organics? And as I was talking with, at the time, my second advisor, we were thinking about all of the different mechanisms that might play a role. And we came to the conclusion that redox plays a very important role. So uranium, zinc, nickel, these are often elements that are going to indicate a change in redox state of a particular environment. So basically how much oxygen is in a system. And so that was very eye-opening and insightful, because oftentimes when you're doing these elemental abundances analysis, most times people really, even though they can get a suite of elements such as what we see here, they often kind of just focus on iron, titanium, silica. Even me in the past, I never would have thought to look at uranium and zinc, etc. And so this provided some agnostic insight into our data set. And what was really cool was just last year, there was a paper at the end of the summer that describes redox-driven mineral and organic associations in Jezero crater on Mars. So I'm not going to say my model basically showing this precisely, but there is some indication that redox really is probably driving a lot of the organic production and preservation associations that we see in particular environments. And so some main conclusions from this first part of the talk is, one, with the unsupervised and supervised learning that I use on this small data set, we kind of provide a proof of concept that, okay, potentially machine learning can be valuable for making predictions of organics, maybe understanding these complex data a little bit more and obtaining insightful relationships. Additionally, this type of data is small, relatively not complex, unimodal, but again, the machine learning framework still works very well, even after some rigorous testing of the performances of the particular models. However, as I kind of alluded to, there's still some limitations. One, the data set is small. We're also only covering a subset, a very small subset of possible analog environments, that being hypersaline lakes or magnesium sulfate and sodium carbonate dominated. So if we really want to improve these, the use of machine learning model in the system, although for this particular system, it's good, we do need to implement more data and testing. And so for parts 2 and 3, I'm going to go through some slightly more complex data sets in a lot of the current work that I'm doing. And I'm kind of, these are going to be a bit shorter than the first part. But some of my current work right now is, one, actually still improving on the first part of the model, so implementing more environments and elemental abundances from various planetary-analog systems, but also developing a novel machine learning framework to investigate multi-scale and multimodal observations. And in parentheses, I'll say medium data, because for this particular work, it's really only looking at two main environments. But one of the main goals for this particular work is, oftentimes with astrobiology research, we can assess an environment from many different aspects. A lot of times, perhaps organic geochemists such as myself might just be looking at lipids and maybe some environmental parameters, maybe some chemistry, but then you have like some cooler people that are also assessing the environment from multiple different levels of remote sensing, such as, again, some of the people in this room, I will not name names. And so for a lot of this work, what we're interested in is using different scales of observation. So something like orbital imagery that we can get from satellites, and also drone imagery, which we can consider to be like sort of this mesoscale observation, and incorporating the in situ observations that I do in the environment. And because of the fact that in situ observations are unfortunately often point focused, whereas remote sensing observations, such as with orbital imagery, you can actually scan really wide swaths of the environment. It might be better if we want to think about planetary mapping of organic molecule space or organic carbon space. And so that's the main goal of this work. And that's actually with in collaboration with some researchers at Dartmouth, Marissa Palusis, and also Will Levitt, who was at Dartmouth but is now at the University of Utah, and then Dr. Emmy Hughes here, and then Dr. Jacob Buffo at Dartmouth. Yeah, he's at Dartmouth too, at Dartmouth College. And so with Marissa and Will, we're mostly characterizing and using the same methodology in the Atacama Desert, whereas with Emmy and Jacob, we are attempting to implement this, and Francis as well in this proposal that we're writing, attempting to implement this in the British Columbia. But for the most part, because most of the work with respect to this multi-scale and multimodal observations is currently being done in the Atacama, I'll focus on that. But as I mentioned, these traditional methods of machine learning, such as random forest, k-nearest neighbor, et cetera, are really good at extracting features from these smaller data sets. They're not so great at extracting features and patterns from more complex data sets, and definitely not imagery. So we have to change our methodology of machine learning approach. And as I mentioned before, maybe neural networks are not always that great because they don't really provide any explainability. We often call neural networks black box models, but we don't know what's actually happening in here. Well, that is the case for artificial neural networks, but recent work, and it's not so recent anymore, it's like 10 years, but I guess it didn't say. It might be the next one. But we can also implement convolutional neural networks, which are a type of neural network that can actually assess imagery and patterns and relationships that are actually spatial, which is really useful. And here again, because I'm mostly focusing on the Atacama Desert, I'm just showing some really nice pictures of how we are sampling in the Atacama Desert. One thing that you'll notice is this is a lot more involved than what I was doing in British Columbia, mostly because the machine learning work with British Columbia was more opportunistic than anything. And I was looking at the data that I already had, but here, because we were already interested in implementing this multi-scale observation, we do a variety of in situ sampling, which you can see here. This is actually a drone image from Alex Morgan. Where's Alex? Somewhere. Alex Morgan, who's done a lot of the drone work for Marissa Palusis. And then we also have some satellite imagery as well. So we have a wealth of data from the Atacama Desert spanning different scales and different modes, that being imagery and geochemical information. And then I have to throw in these pictures here because they're cool. But also, as I mentioned, we are hoping to implement this same methodology and approach in British Columbia with another caveat of potentially implementing this for colder systems. So British Columbia, these hypersaline lakes actually can get very cold in the wintertime and they freeze over. And that's what makes them actually a good analog for Europa. But again, we'll leave that to the experts. I'll stick with Mars for now. But here's Jacob Buffo doing some sampling of the ices. And then here's an awesome picture of Emmy. Is that Bass Lake 1? I think it is. Bass Lake 1 or Bass Lake 2. But hopefully we can implement a similar approach, although it'll be very much more wetter than Atacama. So we'll see how that goes. And so one of the reasons why we choose a convolutional neural network for this particular approach and is a framework that I've been developing with a couple of my students is the fact that the convolutional neural network actually, and again, I won't really go through this entire diagram here, but unlike most machine learning models, convolutional neural networks can actually take information from images, apply what we call convolutional filters, and assess particular spatial relationships, and do that a couple of times. And then we can actually produce like a single vector of all of the spatial relationships from a particular image. And so this is going to be extremely useful when we want to implement these this type of mode of data, that being imagery. But also we have other environmental parameters that we're interested, such as elemental abundances that I mentioned before, but also mineralogy and other spectral reflectance data that we might get in addition to the spatial data from the imagery. And so for part of this particular framework that me and a couple of my undergrad students have been developing is developing what's called a multi-input convolutional neural network, which takes multiple inputs of data, so that being orbital imagery, drone imagery, and then combining that with a random forest model to apply our geochemical and spectral reflectance data. We can then extract those features that I mentioned a couple of slides ago from random forest and combine all of that into a large, basically a large vector of features that incorporates data from the orbital and drone imagery, as well as the geochemical information. And from all of that, we can then potentially make some predictions about things like metabolic diversity, organic carbon content, which includes organic carbon, but also lipid biomarkers, and then also the stable isotopic composition. And so here's the gift that I was looking for. I put out the wrong slide. But the convolutional neural network, this is just a GIF that kind of shows you how it works generally. This is not necessarily the best GIF, but basically you can see if we have some input of images, in this case numbers, these different layers of the convolutional neural network can identify regions that it deems as important for making a distinction for a particular input. And so in this case, we can see that ones are obviously identified by straight lines. We can see curves and fours and zeros. And so we can actually extract that information out and then use our domain knowledge and domain expertise to understand the data better. And so we've actually done this briefly or preliminarily for the Atacama orbital images, and we can actually start to already see important regions and features from the data that it deems as important for predicting the abundance of organic carbon. So for now, I would take these feature maps with a grain of salt, no pun intended, because we still want to add more data. But here we're basically showing that our framework actually does work. And so the main takeaways from this this part of the talk is that, well, when we're working with these multi-scale, multimodal data, CNN random forest framework might offer a way to predict biosignature abundances from these slightly more or maybe more complex data sets than what we saw from the first part, which was just looking at elemental abundances. Again, even though this is more complex and even more data, we're still limited by relatively small data sets required for intensive use of machine learning. And so the final part, which I'll briefly go through, since I know I'm running low on time, is foundation models for astrobiology. So how can we scale this upwards and think about big data? And one of the projects one of the projects that I'm working on is with a group, various people at NASA institutions, as well as universities, and actually Anton as well, is we're thinking of developing an astrobiology foundation model to assess or to use machine learning for perhaps these smaller and disparate data sets. And so I won't go through this image figure in great detail, but one way that you can think of foundation model is kind of like ChatGPT, what a lot of people might be familiar with. So this particularly is not ChatGPT, but ChatGPT is a model that's trained on trillions of different parameters. And we call that sort of a foundation. It's really good for general knowledge. And then with other, let's say, chatbots, they might use aspects of ChatGPT to do some, let's say, downstream tasks. So basically, ChatGPT, it's a model itself, but it also works to alleviate some of the discrepancies in data sizes that you might have with smaller smaller data set, so it allows for fine tuning and deployment. And then with foundation models, because it's often trained on these vast amounts of data, again, like ChatGPT, it might allow us to answer some of these different questions that we might be interested in, such as like, is this lifelike, or is this less lifelike, or more lifelike, etc. And then finally, I will just kind of skip over this, but we're thinking we've been discussing a different roadmap for astrobiology foundation models and how we can implement that. And one of the main takeaways from this is it can really perhaps enable some centralization of data, which is really what we don't have in astrobiology to be able to really use machine learning effectively the way that other fields can do. And then finally, I'll end with some implications and potential future uses, which is once we have models that we're happy with and perhaps have done some rigorous testing with, then we can potentially implement that on, let's say, Martian rovers or orbiters, and start to implement what's called transfer learning. So learning what we've identified on Earth to perhaps create some organic distribution map on Mars or perhaps elsewhere in our solar system. And I'll just end with this takeaway slide, which I briefly discussed, and then some acknowledgements.